Welcome, sir. I request participants not to present their screen or do not present. Please stop presenting.
Good morning, Dr.
Good
Praveen.
morning, sir.
Good morning, Dr. Arvind, sir.
Good morning. Good morning.
Shall we start, sir?
It's a 3 minutes. 3 minutes. We have to wait. It's I think
Okay.
9:58. 2 minutes.
Yes.
आणि बस लेक्चर ऐकायला चांगलं आहे कोरोनावर.
Once again, I welcome all.
एकच तासाचे काय फार नाही.
Participants are requested to mute their mic. Welcome, Honorable Dr. Vaidya, sir.
Okay, thank you, sir. Thank you. Thank you.
Welcome, sir. Shall we start, sir?
Yes, you can.
It's now 10 a.m. I welcome all the participants. A very good morning to all. I, Krishna Bhartakke, welcome all on the behalf of Santral Savarkar Arts, Science and Commerce College Bid for the national webinar on global race to develop COVID-19 vaccine. The webinar is organized by the Department of Microbiology, Santral Savarkar Arts, Science and Commerce College Bid in association with the Microbiology Society India. Dear participants, all of us are experiencing the effects of COVID-19. Very rapidly, the endemic nature of the COVID has become pandemic. The conditions have changed our lifestyle and set new commons. And as a result, we are having the opportunity to be here on the online platform. There are many questions about the rescue from COVID-19, and everyone is expecting their proper answers. In the meantime, we have come across theories and suggestions to avoid the COVID-19 infection. Hereafter, everyone is highly eager to know when a proper vaccine become available to come to combat the disease. As an attempt to make clear the status, strategies, and mechanism of vaccine development, we have decided to organize the expert talk on this burning topic. We are getting much news about COVID vaccine and found that there is a race for its development. All of us are interested to know the facts of this race and to come close to the race or to know the facts of the race. The webinar is organized here. The goal of this webinar is to convey the status of vaccine development to the society through eminent personalities. It is worth to notice a huge response for this webinar. Academicians, students, research scholars, and industry personnel from various colleges, universities, institutions, and industries have joined the webinar. Once again, I welcome all. We hope the webinar will definitely help to resolve the status of the race to develop COVID-19 vaccine. The organizing committee has tried its best to have ideal speakers on this topic. Dear participants, today we have
नमस्कार नमस्कार सर
eminent personalities with
ते
us.
लिंक पाठवले सर हो
Dr. Praveen Alamuri, Director, Product Development and Program Management, Immuneering Corporation, San Diego, California, USA, as a resource person. Professor Dr. M. Deshmukh, the President of Microbiology Society India, is going to deliver keynote address, and Honorable Dr. Hemant Vaidya sir, the Joint Secretary, Bharatiya Shikshan Prasarak Sanstha, Ambazogai, as the President of this webinar. I welcome them all. Dear participants, it's matter of great pleasure to inform the presence of dignitaries for today's webinar. We have with us Honorable Dr. Surendraji Alurkar sir, President, Bharatiya Shikshan Prasarak Sanstha, Ambazogai. Honorable Alurkar sir has always motivated us. I welcome him on the behalf of the organizing committee. Also, we have with us our patrons from Bharatiya Shikshan Prasarak Sanstha, Ambazogai. I welcome them all. Honorable Chandrakant Mule sir, President, College Development Committee of Swatantraveer Savarkar College and Joint Secretary of Bharatiya Shikshan Prasarak Sanstha, Ambazogai, has joined us. I welcome Honorable Mule sir. I also welcome our principal, Honorable Professor Dr. Sanjay Shirodkar sir. All of them have encouraged us for handling the topic to create awareness in this regard. The Microbiology Society that partnered the webinar and academicians, researchers, and students from various social media platforms facilitated to reach the audience and to bring us together from all over the India and abroad. The teachers of Microbiology Society, as well as the teachers from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, and from the Maharashtra have supported us and participated in the activity. I also welcome them and their positive gesture. Now, I request our Honorable Principal, Professor Dr. Sanjay Shirodkar sir, to give his introductory address.
Sir, please unmute yourself. Sir, please unmute yourself. Shirodkar sir, please unmute yourself.
Sir, it's again Mike.
Sir, please unmute yourself.
it okay, I believe?
Yes, now
Namaskar.
you are audible, sir.
Yeah. Namaskar. A warm and healthy good morning to each and everybody from Maharashtra State, India and abroad. I welcome President of today's webinar, Dr. Hemantji Vaidya sir, a well-known gynecologist and also Honorable Joint Secretary of Bharatiya Shikshan Prasarak Sanstha, Ambezogai District Beed of Maharashtra State, India. I welcome Dr. A.M. Deshmukh sir, President, Microbiologist Society, India. Everyone is eager to hear his keynote address. The speaker of today's webinar, Dr. Praveen Almori, Director of Program Management, Director and Program Management, Immunoring Corporation, San Diego, USA. Friends, our college is run by Bharatiya Shikshan Prasarak Sanstha, Ambezogai, BEAT, a reputed institute having a unique perception and goal. The Honorable President of our institute, Dr. Surendraji Allurkar, Honorable Vice President, Shri Radhesamji Lohia, Honorable Secretary, Shri Nitinji Shete, Honorable Joint Secretary, Shri Chandrakantji Mule, Dr. Hemantji Vaidya, Dr. Mrs. Kalpana Tausalkar, Honorable President of Local Unit of BSPS, Shri Govind Prasadji Barshikar, Honorable Secretary of Local Unit B, Shri Milinji Chinspurkar. All these officials, they inspire teaching as well as non-teaching staff to conduct different activities. This support in fostering career-oriented student and academic excellence. Our students are educated to love and respect parents, elders, family, society, and nation. Friends, today, every one of us is very much bothered about Chinese viral invasion after World War II. Now, today we are facing World War SARS-2, that is Severe Acute Respiratory Syndrome 2. The coronavirus is bothering each and everybody at all the world. So many deaths are recorded. Even the weapon could not record so many deaths. Everybody, every scientist is bothered about it. Friends, we will definitely win over this war. The weapon of this war will not be a nuclear weapon. It will not be definitely a nuclear weapon or any other weapon. The weapon of SARS-2 war will be invented by microbiologists. I welcome each and every microbiologist for this. The name of this weapon is vaccine COVID-19. Today, we all are interested to know about latest development of this vaccine for COVID-19. I congratulate Dr. Krishna Bartake, Head Department of Microbiology, a very elegant personality, Shri. I welcome and congratulate Shri Anand Deshpande, a very active personality of Microbiology Department, for their brilliant efforts in conducting this webinar. I am sure this seminar will provide a very rich content to all of us. Thank you very much for attending and participating in.
Thank you very much, sir. Now I request Honorable Dr. Hemantji Vaidya, the President of today's webinar, to give the presidential address. I call my colleague, Mr. Anand Deshpande, sir, to give introduction of Honorable Dr. Hemant Vaidya, sir.
So good morning, everyone. I'm here to introduce Dr. Hemant Vaidya, the President of today's webinar. Dr. Hemant Vasantara resides in Gevrai Taluka of Beed District. He is a joint secretary of Bharatiya Shikshan Prasarak Sanstha, Ambazogai, which is our parent institute, and a renowned medical practitioner in Bid district. Dr. Vaidya has working as a devoted volunteer of Rashtriya Swayamsevak Sangh since he came in contact with the RSS at an early age. I think this is the significant identity of him. Dr. Vaidya has striving for the dissemination of quality education in rural areas and for education to reach the lower sections of the society. He has major contributions in establishing of Chinteshwar Vidyalaya Gevrai as well as the other educational institutes in BSPS Ambazogai. He is associated with many social and cultural activities in Bid district. Being a joint secretary of our institution, he has been constantly encouraging us, motivating us through his work and able guidance. I wish him a very good health ahead and request to deliver the presidential address. Dr. Vaidya sir, please.
Good morning, everybody, and welcome, everybody. We are going to arrange some webinar on microbiology department, which is very needful and which is very essential to update our knowledge and to establish in world. So all my colleagues, I respect to all my colleagues, Mr. A.M. Deshmukh, Praveen sir, Dr. Sanjay Shirodkar sir, Dr. Krishna sir, and Dr. Anand Deshpande sir. And also welcome to my president, Mr. Alurkar sir, my colleagues, Vikas Rauji Dubey sir, Chandrakantji Mule sir, and Mrs. Kalpana Chausarkar madam. At this situation, I am very exciting because this international webinar organized by our colleagues and team. I am thankful to all which effort taken by all my colleagues. Definitely, when we are suffering from this such COVID-19 disease or danger vaccines, we think for what to do and we develop some all these sources and essential condition to prevent this pandemic in the world. And I confidently say the Indian peoples and Indian scientists will succeed in all this situation. Comfortable situation in all necessary good idea about this. And all this discussion and all this content we will find a very fine successful. I also thank to give me some chance for this situation, and I thankful all the team and all the best for the program. Thank you very much. Thank you.
Thank you,
Thank you.
Honorable Dr. Vaidya sir,
Thank you, sir.
for the presidential address. I request Honorable Professor Dr. M. Deshmukh sir, the President of Microbiology Society India, to deliver the keynote address. Mr. Anand Deshpande is going to give introduction of Honorable Professor Dr. M. Deshmukh sir.
I feel privileged to introduce a renowned microbiologist of India, Professor Dr. Arvind Madhavrao Deshmukh, as a keynote speaker of the webinar. Dr. A.M. Deshmukh sir is a founder president of Microbiology Society India and ex-professor and head department of microbiology, Dr. Babasaheb Ambedkar Marathwada University, Subcenter, Osmanabad. His research areas are nanotechnology, biotechnology, solution to e-waste, biodiversity of actinomycetes, biogas, and dye degradation. He was the chief editor of Journal of Microbial World from 1999 to 2015. 17 students has obtained a PhD degree under the guidance of Dr. A.M. Deshmukh sir. Sir has written 4 books and edited 7 books as well. Sir has organized 40 states, national and international levels conferences, seminars, workshops in India and abroad. The countries include Sri Lanka, Bhutan, Mauritius, Dubai, USA, Port Blair, Andaman and Maldives. Deshmukh sir has also worked as a chairman of Board of Studies in Microbiology and a member of RRC, BUTR, environmental committees of many universities and governments. He was invited as a resource person in about 198 conferences and seminars. Dr. Deshmukh sir has 82 publications in 35 international journals to his credit. Sir has at the forefront in creating awareness regarding the use of biofertilizers and pesticides. He has given number of talks on radio and scientific articles in local language Marathi. He has been involved in conducting virtual classes in microbiology since last 5 years. Through this activity, he has arranged lectures of eminent scientists from India and abroad too. Thousands of students are taking advantage of these lectures. During this COVID period, Sir has created general awareness through writing articles in Marathi newspapers and releasing about 15 videos on social media platforms. It is the Microbiology Society India who has written letters timely to Honorable PM, Honorable Home Minister and Honorable CM, Chief Minister, suggesting lockdown for not less than 21 days, importance of herd immunity, care of senior citizens and children, and mass testing, etc. Thus, he played active role to orient the society as a microbiologist. Sir, we feel proud of you. Thank you, sir. Welcome you in the seminar.
Thank you. Thank you very much. At the outset, I have immense pleasure to deliver a keynote address which is organized by Savarkar College, Bid. The reason is that Bid, or from Bid, the Geurai Taluka is my native place. So it is a coincidence today. The Joint Secretary of your Samstha, Dr. Hemant Vaidya, he is from Geurai. I am also originally from Geurai, and we are participating in international webinar in which the participant from Nepal, participant from Nigeria, and participant from every corner of India are attending this webinar. So I'm very thankful to Honorable Principal Shirodkar sir, Dr. Bartakke sir, my PhD student Anant Deshpande sir, and Dr. Hemant Vaidya sir, as well as the chairman of your Samstha, for giving me an opportunity to talk on very, very burning topic of today. Everyone is discussing about the global race in COVID-19 vaccine. Means industrialists start talking about the same. All prime minister and presidents of different countries are talking only of a global race. We common people are also waiting and talking of global race in vaccine preparation. The objectives of every group in the society are different. Let us first see what is the objective of industrialists like a Serum Institute, like a Cadila, or Bharat Biotech. Their objective is to earn the money. The reason is that how much doses of the vaccine will be required throughout the world. 700 crore population, if we consider, the doses required will be minimum 2 doses. You see, minimum 2 doses. So 1,400 crore doses of vaccine will be required. And if we consider only 1,000 rupees price decided by the Serum Institute per dose, or 2,500 rupees price decided by the Cadila, how much is the economy involved in this global race? So you have used a proper word. There is a global race. So everyone is waiting for vaccine, and there is a tremendous race in the preparation of vaccine. So I'm not going to talk in detail. Within 15 to 20 minutes, I will take only review of the present situation of the global race, because after me, Dr. Praveen, he is an authority from California. He is going to talk in detail about the global race on COVID-19 vaccine. But you see, I have told angle of industrialist. Now, what is the angle of prime ministers or president of every country? The prime ministers and presidents of every country are worried about the economy of their country. Economy is hampered. In India, not only in India, in USA, in UK, and in all developed countries, the loss of jobs is huge. And will not cover our economy at least in coming 5 years. That is the situation. They are more worried and they want vaccine because everyone is looking for a vaccine, and today everyone believes that vaccine is probably the solution for this natural calamity. And we common people were fed up with the situation. The reason is that since from 6 months we are sitting at home. You see it. Actually, I am laughing. The situation. Many ladies have told me, sir, from 6 months we have not opened the cupboard of saris. Why? We are not going outside. Many of my friends are telling that we have not dyed our hair dye. We have never used hair dye during 6 months. Why? We are not going outside. Everyone is fed up with the situation. And we believe that the vaccine is the probable solution to solve this problem, this existing problem. Now, I will not go in the history of vaccine.
Please stop
or
presenting.
or the types of vaccine, because there are different types of vaccine. Just one overlook I will tell you. The Serum Institute is producing live vaccine, whereas Bharat Biotech is producing attenuated vaccine, and Cadila is producing mRNA vaccine. What are the differences? Differences are mRNA vaccine, the multiplication is a very fast, so the production is a very simple for mRNA vaccine. So once a Cadila vaccine is a successful, the number of doses, because we are not worried only about the production of vaccine. Second challenge, is number of doses required. As I have told you, the population of the world is a 700, high more than 700 crore population. And at least 2 doses of vaccine will be required. That much doses we required of the vaccine. So probably mRNA vaccine is a faster vaccine. If we succeed in getting mRNA vaccine, we can supply more number of doses, and this natural calamity we can cure in short time. Then, what is the problem of the serum vaccine? The Serum Institute, one of the topmost institute in the world, which is located in Pune, means we should be proud that it is in Maharashtra. This vaccine, being a live vaccine, there are some problems. We have to store in the refrigerator. We have to transport in the cold condition. We have to take many precautions. Meaning is that, what is the challenge? Challenge is that although the vaccine is prepared, transportation of vaccine and administration of vaccine is a future challenge. Again, let me tell you, we hope that vaccine will be ready at least by some institute at the end of November. That is our belief. Mind it well again. We should not forget that it is an experiment and we should never conclude the result before doing the experiment. Whenever we are doing an experiment, result may be positive, may be negative. But in this situation, we are so panicked that we consider the results will be positive and vaccine will be ready in November 8. Again, I am telling you, there are 2 universities in the world which are very famous. One is a WhatsApp university, second is a TV university. They are working like a universities and giving many fake news. Why? They know that public is panic now. And as public is panic, they are giving false information, which is not a true information. So again, let me tell you, although every one of us, because today 700 members throughout India have registered for this webinar. Every one of us is a panic for the vaccine and waiting that when vaccine will come, when it will be injected. Again, you see, if I'm going to talk on that time period required. Why? Because although, means what I'm coming again first, the TV university and WhatsApp universities are giving false information. Don't believe on that. If you want to believe, means find out the fact in the vaccine, go to the lecture of Aadhar Poonawala. It is available on the YouTube. Go to the lecture of director of Bharat Biotech. It is available. You go to the director of Cadila, that is a Patel from Gujarat, Ahmedabad. It is available on YouTube. Or our health minister of India, or Soumya Swaminathan, who is the representative from India in WHO. Ask them about vaccine, they will tell you the real fact. Aadhar Poonawala is every time telling that minimum 2 years will be required to vaccinate Indian society. From today, if we consider 2 years will be required. Because except the Serum Institute throughout the world, throughout the world, there are more than 230 candidates of vaccine. Candidates meaning is that 231 industries throughout the world are working on vaccine. What are the phases of vaccine? First phase is a research. Once research is carried out, characterization of the virus, isolation of virus, physiological, morphological characteristic of virus. The second phase is a preclinical trial. Preclinical trial meaning is that we have to do experiment on the animals. Which animals? The animals which are having immunity close with human being. So monkeys are at a top level. Monkeys resemble with the human being. So monkeys are generally selected for the preclinical trial. So whenever the preclinical trial is carried out throughout the world, more than 13 vaccines are ready after preclinical trial. Then you should submit your results to the WHO, to the FDA, to the ICMR in India. And then they give permission to the clinical trial. And the clinical trials require first phase, second phase, and third phase. So you see how much time generally required for the vaccine preparation? What is our experience of the past many vaccine, because we have more than 50 to 70 vaccine in our hand for various diseases. Minimum time period is a 4 year. Maximum time period is a 40 years. Means the time required minimum 4 years. Then question arises, how we can prepare the vaccine within 1 year and 6 months? The coronavirus study has been carried out 8 months back. So still we require a 6 to 8 months more. But how it is possible within short time? It is possible because WHO has given special permission. WHO has given special permission. Never before. Today they have given a special permission to proceed for clinical trial parallel. Parallel meaning is that whenever you are proceeding for trial first, side by side, you can do the experiment on phase second. Side by side to save the time. So, WHO is also taking effort to save the time. Again, one more limitation in the preparation of vaccine is funds. Because huge funds are required for the preparation of vaccine. And that is a major limitation always during the preparation of any vaccine. But today, in this situation, every government, every NGO is ready to give the funds. So why this vaccine is a rapid? Within 1 year and 6 months, this vaccine will be ready. That is one of the reasons. For this vaccine, there is no limitation of the funds. For this vaccine, phases are allowed to proceed side by side to save the time. And probably by November end, vaccine of only Serum Institute will be ready, and Bharat Biotech, because in India, only 3 candidates are there. One from Pune, one from Chennai, and third from Ahmedabad. Another 2 vaccines will be ready by January 2021. So, 3 vaccines will be ready. There is a race. Now you see again, Serum Institute is the only institute in the world, not only in India. They have started their production. before getting the results. Meaning is that third phase is going on, but they have started the production. And they told that 30 crore doses will be ready at the end of December. You see, 30 crore doses will be ready at the end of December. But again, what are the problems you see? Again, I'm telling global race is there. What is the problem? If we compare the three institutes in India, that is Bharat Biotech, Cadila, and Serum Institute. Serum Institute has made an agreement. Means they are 30 crore doses they cannot give to India. Already they have made an agreement with foreign countries. So 50% of the doses they have to give to the other countries. It means that only 15 crore doses are available to India. They can supply in December or January, they can supply only 15 crore doses to India only. Again, assume that minimum 2 crore doses will be required. Sorry, double dose is required. For every individual, it seems that 2 doses will be required. If we consider the need of 2 doses, minimum 7 crore people from India can be vaccinated within these 3 to 4 months. What is the population of India? It is a 135 crore. Among them, we can vaccinate within 3 to 4 months or 5 months, I will take. Within coming 5 months, we can vaccinate only 7 crores. So what will be the priority? Government will be deciding priority. Government is deciding the priority. What priority they will decide? First, they will give doctors, nurses, soldiers, police. So that will be the priority. And in addition to that, don't forget, there will be the chances of malpractice because human tendency is there. And some rich people will purchase with black money, they can purchase the doses. And some will go to the malpractice also. And only 5 to 6 crore people from India will get vaccinated after 4 to 5 or 6 months. So again, I'm telling you, vaccination is not the solution at present for COVID-19. Because during this 6 months, herd immunity will be automatically developed. So need not to wait for the vaccine. Herd immunity automatically will be developed. So what precautions we have to take? We have to take the precaution that the persons in our family which are old age person or having comorbidity should get protected during these 6 months. During these 6 months, the chances of death, yesterday only there is a report by the Maharashtra government, yesterday only. They have told that 37% deaths are only due to happy hypoxia. Understand? What is the meaning of happy hypoxia? Meaning is that there is a hypoxia in our body. Hypoxia meaning is that reduction of oxygen level in body, but the person is happy. Person is happy meaning is that person do not know externally that oxygen level is reducing in my blood. He is happy and he is not going to the doctor. So I have prepared many videos. The Ananta Deshpande sir has rightly told that I have made many videos for the awareness. I am requesting every time that please, as soon as there is a reduction in oxygen level, consult to the doctor. Don't neglect the disease. That is the only reason for the death of old person or the person of comorbidity. What is our goal? Our goal should not be to avoid the infection. Infection will be there to every human being on the earth. So every one of you and every one of me, means myself also, will be infected in the 6 months, during 6 months by the coronavirus. We should avoid the death. To avoid the infection should not be our goal, to avoid the death. So at the last, I'm going to tell you that there is a global race. Many countries, you see again, just before 15 days, Russia has told, we have prepared a vaccine. What name he has given? Sputnik 5. And the Prime Minister of Russia, he injected that vaccine to his daughter to tell the public that don't worry, the vaccine I'm injecting to my daughter, meaning is that it is a safe. But later on, after 15 days, they accepted, Russia has accepted vaccine is not useful, and they have taken back that vaccine. And WHO every time giving warning, after every 1 month, WHO is giving warning that don't be hurry in preparation of vaccine, otherwise the vaccine may be more dangerous than the disease. There is a possibility live vaccine can revert back. Then what is the efficiency of vaccine? Adhar Punawala is telling in his video, we don't know whether vaccine will work for 1 year, for 2 years, or whether lifelong. We don't know whether enough antibodies will be developed or not. So everything is in dark. But still, human being always lives on hope. And we should have positive thinking in this critical situation. We should not disappoint the public. But at the same time, being educated citizens, we must know the fact. So, although there is a global race, there is a global pressure for the production of vaccine, there is a pressure from society, there is a pressure from industrialists for earning money. Again, I'm telling you, the pressure is for earning money. Any industry, whenever telling that we are going to prepare the vaccine within 3 months, they are earning money by increasing their share value. Actually, vaccine is not going to come in the market within 3 months, but they are earning money because people believe that vaccine will come in the market and their share price increases, and you see they are earning crores of rupees only by the WhatsApp university. So don't believe on WhatsApp, don't believe on TV, go on correct videos of authentic persons. So vaccine will be there, promising vaccine will be there, but it will take minimum 6 months. At least frontier COVID warrior will get protected from the vaccine. Then the persons with a comorbidity will be vaccinated by the government. And lastly, the healthy persons like you and me will be vaccinated. And our number we may come after 1 year, after 2 years. So believe on that. Let us hope for the positive thing. but there is a global race, and I congratulate the Savarkar College for arranging a webinar on an excellent topic. That topic is a need of today. Everyone is worried. So people should know the fact. And now from California, the good scientist is there. He is already involved in research. He will be able to give the information better than me. So I'm thankful again. I express my thanks to all of you. Thank you, sir. Thank you, Bharatakke, sir. Thank you. Thank you again.
Sir, thank you very much. I am very thankful to you for the keynote address and sharing valuable information with us. Now I call upon Dr. Praveen Alamuri to deliver his expert talk. After his expert talk, there will be a short session of question and answers. Now I request Mr. Anand Deshpande sir to give introduction of today's speaker, Dr. Praveen Almuri.
Hello. I feel glad to introduce Dr. Praveen Almuri as the speaker of this webinar. Dr. Praveen is a native of Hyderabad and now settled in US. Pursue career in research. Dr. Praveen conducted his doctoral thesis research at the University of Georgia. His work was on understanding oxidative stress resistance mechanism in the human gastric pathogen Helicobacter pylori. He discovered and characterized a new class of methionine sulfoxide reductase enzyme, methionines in proteins, and helps the bacterium minimize oxidation. Praveen received his postdoctoral training at the University of Michigan Medical School. He discovered a novel cytotransporter protein in human uropathogen Proteus mirabilis. He characterized three previously unknown cytotransporter proteins in Proteus and demonstrated possible use of inactive toxin protein as subunit toxin candidate. He published a number of research articles on his work. One of the manuscripts was recognized in Microbiology's Faculty of 100 in 2008. He co-authored 7 research articles and is one of the inventor on a patent. From 2008 to 17, Praveen worked in various capacities as program director, program manager, etc. in Arizona State University. He worked in the Center of Infectious Diseases and Vaccinology under the world-renowned bacteriologists. He received his MBA in 2012 and won an international business plan competition in Windsor, Canada, proposing development of the novel seasonal flu vaccine that was being developed at ASU. Currently, Praveen is serving as a director, product development and program management in Immuneering Corporation and coordinating small molecule-based drug development programs aimed at traveling solid tumors. I welcome you, sir, and I request you to deliver the speech. Thank you, sir.
Thank you, everyone. Honorable society members, Professor Vaidya, Professor Deshmukh, Professor Shirokar, and Anand Deshpande. Thank you very much for welcoming me. It's really an honor, and happy Teachers' Day, belated Teachers' Day to you all. You all are really serving in a noble cause as teachers. My talk today is going to be in 2 parts. The first part is going to be based on the development of COVID vaccine and where we are in a global stage in developing various candidate vaccines, as Dr. Deshmukh had suggested. And the second part of the talk, provided the time permitting, I would like to talk a little bit on salmonella vaccine development. I played a very minor role in the 7 years of my stay at Arizona State University in developing certain candidate vaccines for various infectious diseases. In the first part of the talk, COVID vaccine, I haven't really worked much on it, but I've been always curious as to how the various companies and research institutes have been developing this. So I did a detailed research survey, and I'm presenting some facts that I learned and some from the latest literature that could enlighten all of us in trying to understand where we are in this global race. So can you guys see my screen and can you hear me properly?
Sir, you are audible, but we are not able to see your screen.
Yes, yes.
Okay.
Sir, we are able to see here.
Yes, please continue, sir.
Okay.
Let's continue.
Thank you. So I call it finding the right job for the CV, because CV stands for COVID vaccine, and there are so many applicants, as Dr. Deshmukh had said. There are close to 180 vaccines all over the world that are really racing towards reaching the common man. First, let's try to understand what the coronavirus looks like. The coronavirus belongs to the family Coronaviridae and the subfamily Coronavirinae. It's a beautiful round-shaped virus with petal-like projections made up of this spike protein. And it has an envelope structure around it, and a nucleocapsid protein and a hemagglutinin esterase and membrane proteins. There are just like 5 or 6 proteins that make up the coronavirus. The virus has a genome, it's an RNA virus. All coronaviruses are RNA viruses. The SARS-CoV virus that infected a good part of the world in 2002 belongs to the same beta family of coronaviruses. And the MERS virus, the Middle Eastern Respiratory Syndrome virus, which has a mortality rate of about 30% in mid-2000s, like 2008 to 2009, that also belongs to the same family. We are just fortunate that SARS-CoV-2, the current pandemic virus, has a mortality rate of less than 3%. So if 100 people get infected, there's only a chance of only 3 people succumbing to death of this virus, which is actually a fortunate thing, considering the recent spread of Ebola, considering the recent spread of MERS virus and all, with a much higher and larger mortality rate. So the genome of the coronavirus is RNA. It's around 30 kilobase pairs. It is 76% identical to the SARS-CoV virus that occurred in early 2000s. That's one of the reasons why we are able to make the progress we are making in developing this vaccine, because we already know a lot of information about a similar vaccine that we have encountered about 7 or 8 years ago. We are using that information to move forward in developing the current vaccine. And you all might have read that part of this coronavirus must have occurred transmitted from bats. The virus that is the coronavirus in bats is about 93% identical genetically to the virus that is actually infecting us currently. So how does this virus actually enter the human cell? I'll be focusing more on the vaccine development, but it is probably very useful for everyone, including me, to try to understand how it enters the human cell, how it causes disease in a very nutshell, in a very simple synopsis. So this is the virus cell, which has the antennae kind of these rose petals kind of spike protein. The spike protein attaches any lung or kidney or heart or intestinal epithelial cell. And using some proteases, this virus enters the human cell, gets into a vacuole. Any virus or bacterium that enters an epithelial cell usually is surrounded by a vacuole kind of a structure. That's a protection mechanism by our body. So that's called an endosome. The endosome, the pH drops and the virus breaks down, releasing its RNA. The RNA, it's an RNA virus, I told you. the RNA quickly gets translated by its own ribosomal proteins, and the 5 proteins that are important for the production of the virus are formed. The DNA RNA complex that actually, the RNA polymerase complex that is required for further multiplication of these copies of RNA is made. Here, these are the subgenomic RNAs that are of the virus that are made. The virus completely hijacks the cellular machinery, and then the virus enters the nucleus of the epithelial cell, where the translation takes place in the nucleus of the eukaryotic cell. Then after the proteins are assembled, after the protein polypeptides are made, the virus is assembled again into the endosome, and finally it is released and matured. This is a simple life cycle of the virus. I'm just simplifying it to the best possible in the respect of the time. The recent drug produced by called remdesivir, that is produced by Gilead, this is how it acts. It actually stops the virus for RNA polymerase complex from forming and making multiple copies of the RNA here. So once the virus can get in, but it cannot replicate because its complex is shut down by the drug. It also shuts down any protein synthesis because it totally closes the macromolecular complex of the virus, and that's how it totally arrests any further infection from spreading to other tissues. So how does this, Dr. Deshmukh very elegantly explained how the breathing hypoxia happens, like we are not able to breathe properly when there is severe infection of the virus. So when the virus attaches to the cell, it attaches to a protein called ACE2. It's a small receptor on epithelial cells. Earlier, it used to be known to be only on lungs. Now, because of COVID, doctors and scientists have found that this ACE2 receptor is found on kidney, intestine, liver, and many other organs in the body. Once the ACE2 receptor receives, it's almost like a gatekeeper for the virus. It actually attaches to the virus, sucks the virus into the cell, and once the virus gets into the cell, it creates inflammation. So the body gets disturbed. All the immune system gets terribly activated. It's almost like a panic situation inside the body. When the inflammation occurs, a lot of fluid edema, or like a lot of fluid is accumulated in the lung. So that stress is what causes from stopping us from breathing peacefully. When that stress occurs, ACE2 protein further upregulates because of the stress, and it further invites more viruses. So it's like a vicious cycle. So that is what happens in a simple infection cycle of COVID-19. So that's why they use corticosteroids to drop this inflammation rate, to drop this ACE2 production by the cytokines. They stop it so that this inflammation doesn't occur. So here is the picture down here is like a healthy lung sac can easily exchange oxygen and carbon dioxide, whereas an inflamed lung that is infected by pneumonia or COVID or flu, anything, it creates a lot of water inside the lung cell, and you cannot exchange oxygen and carbon dioxide. That's what causes us from this breathing problem. And so people who've been following some literature on COVID-19 must have heard about the term called cytokine storm. It's like a flood of immune cells attacking our own tissue. How does it happen? So when the virus comes in, or any bacterium or virus comes in and attaches to any immune cell, the T lymphocytes and the neutrophils immediately attack the virus. They inactivate the virus, and then the neutrophils come in and clean up the mess. It's almost like the T cells come, kill the virus, all the mess is cleared of the neutrophils, and the rest of the immune system is shut down. So that they say that the T effector cells or the B cells or T regulatory cells, they stop the immune system to further getting too activated. That happens in a healthy lung, like a healthy infection, a regular viral or a bacterial infection. In case of COVID-19, the immune system is always tricked out. The immune system is not able to recognize virus, and the virus is very easily being escaped by the virus is able to escape the immune system. So the initial attack of neutrophils or T cells is not happening. the virus skips through the initial steps and creates a panic in the immune system. There is like T effector cells, macrophages, neutrophils, a huge pool of cytokines like interleukin 2, 6, 7, 8, all they actually make a total mess of the tissue. So what happens is it almost like creates like a bomb blast inside the lung or kidney or heart, because the immune cells don't know what to do. They just kind of all come together and they damage the tissue. So if a regular virus comes, it's actually our immune system is supposed to, it's like a quick analogy for you to understand. The immune system should attack like a sniper. The bullet should directly go hit the virus, and that's it, no damage to the tissue. But in times of, in case of COVID-19, it turns out like a bomb blast. So the virus might get killed, but there is a lot of damage that happens to the body. And that's where you get these organ failures like breathing problem, heart failures, intestinal problem, diarrhea, all these things happen because of our immune system hurting us. That's what is just wanted to set a platform for you to understand where all what the situation in the body is when the virus attacks. So we all know there is close to 26 million cases of COVID-19 currently registered, and 873,000 people have succumbed to death. And this is the latest data from like end of July by WHO. So as the scientists before told us, there is a very swift response both from corporate sector and from the scientific community. There are close to 700 different attempts being made, like 700 candidates or 700 treatment regimens currently being administered. So those fall under vaccines. There are 181 vaccine candidates currently, 191 antiviral drugs, and total 335 different treatments. Here I can say even taking a crocin is also a treatment. So when I say 335 treatments, taking a crocin or a headache tablet is also considered a treatment for COVID-19, or taking a BP tablet, because people having blood pressure, the medicine they take for blood pressure suppresses the ACE2 protein on the lungs and the liver. So the virus doesn't easily enter into their tissue. So people who take blood pressure are slightly more resistant to this infection than the normal public. It's kind of a good side effect to have. So there are a lot of countries, as was mentioned, that are in this race of trying to make an impact globally. So this chart here shows you in January there were 3 vaccines that started developing, and as we move down the line to August, you see the bars increase over here. There are totally 179 to 181 candidate vaccines currently under pursuit at various stages. Some of them are probably trying to test in the laboratory or in animals, and some of them are probably in the phase 1, 2, or 3 clinical trials. But overall, there are 179 vaccine candidates currently being pursued. So this panel on the top that gives you that antivirals that were all total 335 or 200 antivirals that were created, 61% of them were created only for COVID-19. 27% of them were created for something else, but they are being used for COVID-19 with slight modification. And repurposed is they changed the target and they are being used for COVID-19. But when it comes to vaccines, 95% of the close to 200 vaccines that are being developed are purely for COVID-19 virus. There are 3% to 2% that are actually being developed for something else, but because there are certain merits in that vaccine that will help us actually use it against COVID-19. And different technologies here, I list few of the technologies that are being used. There could be a protein-based vaccine, a viral-based vaccine, RNA vaccine, DNA vaccine, or immunotherapy. That means a certain person gets infected with COVID-19, hospitalized, the person gets released, they can take the plasma, the blood, they draw blood from the person, isolate the plasma, and then they are trying to immunize the other person because there is antibodies in this person that got hospitalized, that got infected, and that got cured. So there must be some antibodies, some component in the blood that might be used for some other person. Well, that is being debated whether or not it's a good thing to do or not. But let the entire scientific community decide on whether or not to go that route. But so this is a nice cartoon I found online, and I thought this was really very apt for the discussion. So we all know the tortoise walks very slow. So that is the perception of general public perception of the COVID-19. Everyone is so panicked that they think that the scientific community is moving very slow in trying to address this issue. But a snail, which actually moves much slower than the tortoise, feels that the tortoise actually is moving fast. So that's the scientific community. We are moving at such a rapid pace without even noticing that we don't even know how well these vaccines will work, how much harm they can do. So the scientists are excited to actually address the issues of the public pandemonium, but at the same time, they're generally cautiously optimistic because we really don't know. It's a black box until maybe 2 or 3 years pass on. So as I said, speed is always relative. Someone going on the bicycle always thinks the car is faster. Someone going on a bus thinks the car is slower. So that's where we are with the COVID-19 as well. So this is where I'll dig into the topic of various stages of vaccine development and where we are. So the vaccine, any drug development doesn't have to be vaccine. Any drug development goes through these series of phases. A proof of concept that you have a candidate, you have a target, the candidate can actually interact with the target and do its job in the lab. That's your proof of concept. Preclinical is once it is positive in the tissue culture, in the Petri dish, you take it to animals. It can be rabbits, mouse, ferrets, anything, guinea pigs. That is your preclinical. Phase 1 is a small set of population just to test that the vaccine is not doing any harm. Phase 2 is increasing the population. Phase 3 is giving to actual patients and a wide variety of population demographics to see how well it is actually being captured. And this is where most of the vaccines are generally opened for first line of defense people, like military, doctors, and all. So let's go over the list of candidate vaccines currently in the top of the race. Again, I claim here, this is not a complete list by any means. It's not possible to cover 180 vaccines by any means. I just chose like 2 or 3 vaccines from each technology that are in the farther end of the race, so that we can really know, we can feel optimistic and better about how things are, to hope that one of these vaccines pans out. So the first is the Serum Institute of India and the Vector Project, Virus Project Management, Vaccine Project Management in Germany is partnering with Serum Institute of India. That's the altered state of BCG vaccine. Then the novel mRNA vaccine by Moderna Biotech. Dr. Deshmukh sir was talking about how the share prices are increasing. Moderna share price was $6 in January. It is $65 today because they've entered phase 3 clinical trial, and it's one of the most promising vaccines you might see in December or January. So that's the impact on the share price, if anyone is interested in stocks. So the other thing is the measles vaccine by Egypt. And there is a series of attenuated vaccines like the coronavirus. They actually modify the vaccine virus in such a way that it cannot produce any disease, but it can stimulate the immune system so that the immune system gets activated. That's the function of the attenuated or inactive vaccine. Then the DNA-based vaccines, Zydus Cadila and the AstraZeneca, and Novavax Takeda vaccine. So these are the vaccines I will try to cover in the given time. What is the technology behind each of these vaccines and at what stage they are? If there are any questions, please speak up. And if you can't understand, if I'm going too fast, please speak up and I can slow down or I can repeat myself. If I don't know any questions, I'll be happy to look into the literature and let you all know later. Okay. So how vaccines work, I'm sure everyone knows it. I'm just kind of bringing everyone to the same speed. Vaccines are nothing but a component of a virus or a bacterium or a whole bacterium or a whole virus that is weakened, that when given to a human or an animal can stimulate immune system so that it gets prepared with the defense cells in such a way that when the person gets in close contact with that pathogen, virus or bacterium in the future, the person will not get an infection. That is a simple principle of vaccine. And for the vaccine to work, the vaccine has to be safe. It shouldn't have any side effects. And the vaccine has to give maximal antibody response or T cell response so that the individual who has taken the vaccine is prepared to fight the infection even years down the line. We all take TB vaccine, polio vaccine when we are kids. That is a lifelong immunity. TB vaccine is a 13-year immunity. Polio vaccine, chicken pox, and all are lifelong immunity. Whooping cough is a lifelong immunity. So, can you guys see my screen?
Participants are requested not to present their screen. Mr. Prashant Kumar, please stop presenting the screen.
Because someone is presenting their own screen.
Mr. Prashant Kumar, please stop presenting the screen.
Okay, can you see it now?
Yes, sir. Fine.
Okay. So that's how the vaccines work. So let's go through each, like a sample vaccine for each of the technologies. The first vaccine is the AstraZeneca vaccine, which is partnered with the University of Oxford, and it received close to $1 billion from the USA government under their war program. So it's named Covishield. The clinical trial base is in Chennai. So it is a recombinant adenovirus vaccine. What does that mean? Adenovirus, we all must have encountered adenovirus in our lives almost like 10 or 20 times. Simple cough, simple sneeze, simple cold, or a simple diarrhea. All these are symptoms of one of rhinovirus or adenovirus. So we all are immune to adenovirus. Adenovirus can't really harm us as much. So what AstraZeneca did is their technology is they took the adenovirus from chimpanzees and they actually inserted the gene encoding the spike protein. In the initial introduction, as I said, spike protein is very important. Those are the petal-like antennae structures that attach to the epithelial cells, and they will pull the virus. They will push the virus inside the cell. So all the vaccines right now are targeting the spike protein. They think that if the spike protein can be blocked, the virus cannot enter the epithelial cell, and you won't cause an infection. So 90% of the vaccines that I am currently telling you are targeted at the spike protein. Okay, so same thing with this one. So adenovirus is a double-stranded DNA virus. So they take the mRNA component of the spike protein, the mRNA fragment that codes for the spike protein. They take the cDNA, the complement DNA of it, and they clone it in the double-stranded DNA of adenovirus. So what do you have here in this picture? You can see you have a regular adenovirus. The adenovirus genome is in red in color, and they insert the gene for the spike protein in green. So when the adenovirus enters the cells, it just multiplies and replicates in the same fashion. It produces its own protein inside the human cell. At the same time, it produces the green spike protein also. Without the human cell doesn't know it's producing spike protein. The adenovirus machinery just takes on. When that spike protein is seen by the antigen presenting cells like macrophages, dendritic cells, and all, they process the protein and they stimulate the immune response. So that is the recombinant adenovirus-based DNA vaccine vector. It's like basically it's a DNA vaccine vector shuttled in, carried by the recombinant adenovirus.
Sir, your screen is not visible.
Yes, sir. Your screen is not visible, sir.
How about now?
No.
Yeah, it's going on, sir. Thank you, sir. Thank you.
Okay. So let me go back. I'm sorry, I didn't realize that your screen is not visible. So the adenovirus is a double-stranded DNA virus. What AstraZeneca did is they took the gene that encodes for the spike protein of the virus, of the coronavirus. They inserted the gene into the adenovirus genome, and the adenovirus is injected into the humans, animals, wherever they want to immunize. So when the adenovirus replicates, with it, the spike protein is also, the product is also made, the spike protein is also made. It is taken up by the antigen presenting cells, it is processed, and it is shown out, it is projected out as an antigen in the human body. So when the immune system recognizes the adenovirus and other particles, it also recognizes the spike protein, and an immune response is triggered. And that's how the person gets immunized against the coronavirus using the recombinant adenovirus vaccine. So here is the data that was just published last month from the clinical trial. The red dots here are the samples from patients immunized with this actual vaccine containing the spike protein. The blue dots here are the negative control. As you all know, every experiment needs a negative control to show how good your experiment is working. So the blue bars are green fluorescent protein from Drosophila, which is actually going through the same cycle of the adenovirus recombination vaccine vector. So that is your negative control. So two populations are immunized. One population gets the actual vaccine. One population gets the duplicate vaccine, that's in the blue. The population getting the actual vaccine, you can see the antibody titers are very high. The immune system recognizes the spike protein. So when they take the blood from the people immunized with the recombinant vaccine, and they do an ELISA test, we all know what an ELISA test is. It's a protein-protein sandwich test. So the serum taken from the immunized humans can recognize the spike protein. That means the vaccine actually, in theory, it worked in its clinical trial. Not only did the immune system get triggered, the immune cells that were produced during this vaccine can actually see the virus, and they can kill the virus in the petri dish. So they have a neutralizing, those are called neutralizing titers. So basically the AstraZeneca, the Covishield vaccine was successful in terms of their clinical trial strategy. There were no side effects. They got good antibody titers, and the antibodies can actually kill the viruses in the lab. And this is just basically giving data about how well. So once these monkeys were actually immunized, and when they're challenged, their lungs were protected when their coronavirus was they were infected with coronavirus, and only the nasal swabs had very little virus in them. Their lungs were clear of coronavirus, and their intestines were clear of the coronavirus. So the DNA, the recombinant adenovirus, actually worked very well in monkeys. This virus is in the phase 2 clinical trial right now. They haven't really published any of their phase 1 or phase 2 data. I searched online, but I couldn't find it. But their preclinical data is pretty strong. This is the Russian vaccine that the whole world, sorry. This is the Russian vaccine the whole world was talking about 2 weeks ago. It is again an adenovirus-based vaccine, just the one that I explained you 2 minutes ago, where the spike protein is cloned in the adenovirus DNA. There are 2 different types of adenovirus viruses they used, and they used a prime boost regimen. That means, like Dr. Deshmukh said, there are 2 doses required. One is the prime, and the other is the push, the boost mechanism. So this vaccine is currently in phase 3. Most likely, it is possible that they will pull out of the race, because the data is not really clear. But from what they published, this is what I could gather. Whether the vaccine was stored in a frozen condition, or whether the vaccine is stored in a freeze-dried condition, like a powder, frozen powder, when it is regenerated and immunized in humans, you could see very strong antibody response and very strong neutralizing antibodies. That means the serum taken from patients, when you infect in the virus petri dish, that serum taken from the patients can actually kill the virus. So they show pretty strong data, but I'm not really sure why or where they have really lacked proper evidence that they're about to pull out of the race. In fact, if you look at it, if you give both these vaccines together, they make two different kinds of vaccines. If they give two vaccines together, you see the titers are actually skyrocketed. They're much better than these panels. Can you guys see my mouse pointer? Okay. So these titers are much higher than the individual titers. This is the serum from a patient who is hospitalized, who recovered from infection. These yellow dots are the serum from the patients. In fact, the immunized people had stronger antibody response than actually the patients who suffered through the infection. So which is actually a very good news if this vaccine really, if it works. This is the Cadila vaccine that Dr. Deshmukh had mentioned. So this is a plasmid DNA vaccine. It's the most simplest form of vaccine that you can get. We should really hope and pray that this vaccine is successful in India, because many of the protein vaccines or many of the live vaccines require cold storage and cold chain. That means they have to be either frozen or they have to be transported and packed in low temperatures to the nook and corner of the country. And for our economy, that's a huge challenge to face. I mean, to go to every nook and corner of every village and district in a cold storage, it's just not practically possible. But a DNA vaccine is very robust. You can actually save it in 25, 30 degrees and nothing will happen to it. So let's see how this vaccine was created. So they took again the spike protein. This is the crystal structure model of the spike protein. They cloned this protein on a plasmid. Plasmid is a self-replicating molecule of circular molecule of a DNA. They cloned it in, they mix it with an adjuvant, and they inject into mice, rabbits, pigs, and all to test. And they saw a very robust immune response. Unfortunately, again, there was no published data on Zydus Cadila. There were only newsletters published by Cadila and Zydus on their websites. So they just presented their technology, and they said in phase 1 and phase 2, there were no negative effects seen, and the vaccine could actually induce very strong immune response. So this is in close to phase 3 clinical trial right now. And let's hope that, I mean, of all the vaccines, if I really pray that works, should be this one. Because, I mean, not only India, many other economically challenged countries can actually benefit from this kind of vaccine more than anyone else. So as I say, phase 1 is done and they are entering phase 2. Or actually, they are almost about to complete phase 2 on this. And this is the Serum Institute vaccine that Dr. Deshmukh mentioned. This is actually not a COVID vaccine. This vaccine was created to better fight tuberculosis. Let me explain you. We all know when we were kids, we got the BCG vaccination on our arms, right? The big tika that we get on your shoulder here. That's a BCG vaccine. BCG vaccine is an inactivated form of a cow TB vaccine. So like they take the inactivated form of a TB strain that infects cows, and they vaccinated all of us. But the challenge with the BCG vaccine is after the age of 14 or 15, it is useless. So pretty much all of us, although we were vaccinated with BCG, we all are equally susceptible to getting tuberculosis. So the vaccine project management started this project to make a better BCG vaccine. They actually took BCG vaccine, they took out the urease protein out of the vaccine strain, and they inserted a listeria toxin inside it, a toxoid gene inside it, so that it can go and stimulate the immune response in the humans. And they are trying to project this use this for COVID vaccine, because TB also causes lung infection, fluid accumulation in the lung, and also causes breathing problem in humans, right? So what they are thinking is using this BCG modified vaccine for COVID will help at least reduce the level of infection. It may not be able to prevent anyone from having an infection. People might still get the infection, but they might be very easily cured. So I call it more of a therapeutic vaccine rather than a prophylactic vaccine. So there are two kinds. Prophylactic is a preventive vaccine, which will stop or help you prevent getting infected. Therapeutic is the person gets infected, but the vaccine would actually help you get rid of the infection, just like a drug. So that is the principle behind this BCG Serum Institute's BPM 1002 vaccine. So here the bacterium enters into an antigen presenting cell. The bacterium is degraded, but particles of the bacterium are shown out, and they stimulate an immune response. This immune response is useful for any viral particle or any bacterial particle. It gives a broad spectrum immunity against severe infections in the lungs. That's what this pretty much does. It's not a purely, I wouldn't call it a purely coronavirus vaccine. But this again is very useful because it will help from susceptible population like the older people or the people with some underlying conditions like some sort of a blood pressure or some sort of a lung infection from really dying out of infection, because this will help you come out of the infection much faster. And this is the Moderna vaccine. So we covered the recombinant adenovirus vaccine, the Russian vaccine, the AstraZeneca vaccine, and the Johnson & Johnson vaccine is also very similar to the Russian vaccine and the AstraZeneca vaccine. And then we went through the plasmid DNA vaccine by Zydus Cadila. Those were the DNA vaccine family or recombinant virus or DNA vaccine family of vaccines. And then we had this is called, I call it a genetically engineered bacterial vaccine for COVID-19. This is kind of a new class of vaccine from Serum Institute of India. Now we'll move to the most kind of, the kind of, I would say, cutting edge technology in producing or developing COVID-19 vaccine. I will stop my talk for 1 minute and see if anyone has any questions, because there was a lot of technical details that were provided. So if anyone has lost me or I was too fast in explaining something, I'll be happy to kind of repeat what I said if there is if someone lost me in any kind. Okay? You can unmute yourself and ask any question or ask me to repeat anything.
Hello, sir.
Yeah.
Sir, how nanoparticles are useful in the treatment of COVID-19?
I haven't come to that part yet. I will explain that on Moderna vaccine principle. But if you have any questions on the recombinant adenovirus or the plasmid DNA or the Serum Institute vaccine, I'll be more than happy to answer those questions. I'm going into the nanoparticle thing right now. Okay? So let's resume. You guys can see my screen, right?
Yes.
Okay. So the Moderna vaccine is an mRNA vaccine. So as you know, the coronavirus is an RNA vaccine. So what they did was, so Moderna has this in-house technology. It's like their technology, like Microsoft has their Microsoft Word, PowerPoint, and Excel. Moderna has their own home technology called the lipid nanoparticle. It's like a lipid bag. It's almost like a plastic polythene bag that can cover over the virus or RNA or a protein or a DNA. So this bag is their technology. That's what they are actually making their mark in the society right now. So what is this lipid bag or the lipid nanoparticle? It's like a small cholesterol-based or a lipid or a soap-based bubble that they create with lipid on the top, like fat or cholesterol on the top. So there is a two-layer, there is a surface layer outside, which is hydrophobic, which can attract water. The lipid inside, the black lines that you can see inside here, it is hydrophobic. It is oily in nature. It repels water. And inside the third layer, the circle I see here, my mouse is showing you, is the active kind of container where you can put the antigen. You can put a protein, you can put an RNA, or you can put a small virus in it. So it's like a bubble. So they created the bubble. The importance of this bubble is it is harmless. It can carry DNA or RNA or a protein, and it is self-adjuvant. Like, when you need a vaccine, you also need an adjuvant. So it has an alum or a soap-based adjuvant created inside it. So it's all in one small bubble. So once this bubble, this lipid emulsion is created, they actually take RNA of the vaccine, of the coronavirus, create a bubble. Now you have a bubble and an RNA inside. This is injected in all the areas, or select areas of lymph nodes in our body. Lymph nodes are almost like immune banks. Those are like reservoirs of immune cells in our body, right? Lymph nodes. There are several lymph nodes in our body. So this emulsion of RNA and bubble is injected either via intraperitoneally, intravenously, or intramuscularly in various lymph nodes of our body. And then once the lipid nanoparticle enters the cell, it bursts out. The RNA is liberated. The proteins are made, they are processed, and the immune system will know that the virus has invaded by these proteins, and the immune system gets triggered, and the protection is conferred in the human. That's how this whole cycle works like. So once the RNA is released, the human cell thinks that it is its own RNA, and it starts making the proteins, the viral proteins. Once the proteins are made, the immune system sees that those are actually foreign proteins, and the immune system gets triggered and neutralizes the virus. That is the whole principle or the hope behind this Moderna's mRNA lipid nanoparticle technology. Pfizer also has a similar vaccine. Pfizer also got close to $1 billion from US government to develop this vaccine. Pfizer actually bought this lipid nanoparticle technology from a European firm called BioNTech. So Pfizer and BioNTech partnered. They are also creating a very similar technology for coronavirus vaccine, and that is called BNT162. So it's very similar. Let's look at the clinical trial phase 1 data. So they actually immunized humans with 25 micrograms or 100 micrograms or 250 micrograms of this lipid emulsion. And as you can see, after 50 days after immunization, the antibody titers are so high. So the antibody levels, the virus-specific antibody levels in the human body are really high, which is a good news. So the vaccine was delivered, the proteins were made, our immune system recognized it, it produced an immune response, and the immune response can be calculated or quantified in the lab against a viral protein. So it actually could recognize various parts of the spike protein. The spike protein is really long. It's 120 kilodaltons long protein. So it recognizes various parts of the spike protein. And also neutralization titer. That means these antibodies, when you take and put it in a petri dish containing virus culture, it can actually kill the virus. So it's a very strong neutralizing antibody titers were generated in this phase 1 and phase 2 clinical trial. And Moderna is actually they should have started their phase 3 clinical trial on August 26th, but they wanted to actually enroll people who have HIV infection also. And they were actually waiting for the permission. They most likely be starting this phase 3 clinical trial in the second week of September, or maybe sometime around that time. And why is it? So that's the beauty of various stages of clinical trials. So whenever in the drug or vaccine development process, when it hits phase 2 or phase 3, you can actually enroll people with some pre-existing conditions, like some infection, like HIV infection, from some TB infection or something. So you are actually bringing all kind of population into your statistics and trying to see how effective your vaccine is in a varied population. So that's how the development cycle goes in vaccine development. Initially, you take phase 1, you take just 100 or 50 people. Phase 2, you take larger number of people. Phase 3, you take people with disease conditions also. So Moderna vaccine is a successful vaccine in phase 2 clinical trials. And this is a very interesting vaccine that is being developed by Novavax and partnering with a Japanese pharmaceutical company called Takeda. which has a huge presence in the US also. So this is very similar to again the nanoparticle kind what I talked about. So here the spike protein is made, and the spike protein is put in a matrix of cholesterol and lipid matrix, very similar to the lipid nanoparticle. So that matrix M. So here is the picture of electron micrograph of it here. My mouse is showing the electron micrograph here. So the matrix has the spike protein. They inject this matrix with the spike protein into humans, and you get antibody titers. As you can see here, people immunized with this vaccine showed pretty robust antibody titers that can recognize the spike protein in the lab. So these are like very strong titers here, and also very nice neutralization. Again, the same principle. Just having a strong antibody response is not sufficient to tell you that the vaccine is successful. Those antibodies should be able to recognize the virus in the lab and kill the virus, neutralize the virus. It's only then you will know that it can do a similar thing inside the human body as well. That is the whole cycle of saying, of calling a vaccine successful. Here they are actually seeing if they are comparing it with the titers, neutralization titers across the board, like, depending upon how much of the antigen you're providing, the titers keep on increasing here like a staircase. This increase is much better. The humans which are using, which are getting this vaccine, the antibody titers are much stronger than people who actually got infected outside in the general population. So if you take their serum and test in that neutralization assay, it's much weaker than the vaccine that Novavax has created. So that's some good news there. And there is a lot of hope that there will be herd immunity generally, because your neighbor gets infected and the neighbor will not get infected once again. So you're almost like creating a social barrier. But the problem with achieving herd immunity with coronavirus is the antibodies that are actually created in a patient are not long-lived. So a person who is getting infected generates the antibody, but 3 or 4 months down the line, the person is getting reinfected. That means the antibodies that the person got in the first infection, they are not living for 2 or 3 years or 10 years like our TB vaccine or polio vaccine we got when we were kids. That's a lifelong immunity. Coronavirus infection is not providing a lifelong immunity to people. So herd immunity is, there are still some kind of debate in the scientific community whether a herd immunity is indeed possible at all. So for the herd immunity, my neighbor should, unfortunately, if my neighbor gets infected, he or she should never get infected for me to get the herd immunity benefit of the neighbor getting infected. And that's not happening. And that's kind of a little bit debatable right now. So this is a good sign that an infected patient serum is weaker than the vaccine, the serum from a vaccinated person. And when they compared the Novavax with Moderna vaccine or Pfizer vaccine or AstraZeneca vaccine, Novavax, as you can see the blue bars here, I'm sorry, this picture is not clear even in the manuscript that I got. So the Novavax bars are much higher. I mean, you can visually see it, that this response is much strong. Sorry, this response is much stronger than for any other company. So Novavax can be pretty strong, but at the same time, I come back again to how easy or how difficult would it be for such kind of a high technology vaccine to be distributed in a country like India. This is a lipid emulsion, protein in lipid emulsion has to be stored very carefully, has to be administered very carefully in the public. So something like India or other developing economies might actually suffer in terms of administration or storage of this vaccine, because the cost of administering, storing might be more than the cost of acquiring this vaccine to begin with. But let's hope that something gets successful to begin with. And the last piece of evidence I have is the Sinopharma and Wuhan Institute of Virology. Wuhan, as you know, was the epicenter, the ground zero of the coronavirus pandemic. It's in the meat market of Wuhan province where the virus was detected. So the Sinopharma and Wuhan Institute of Virology, they actually isolated the virus from a patient. The strain was called Wuhan Institute of Virology Z404. The virus was passaged several times in tissue culture to weaken it. So the way the virus passaging and weakening, the best example is you buy a new pair of shoes, you walk hundreds of kilometers in it. After a year or two, the sole gets totally smoothened, right? It doesn't have the same grip to the shoe. Similarly, when you take a virulent vaccine and you actually go over and over, you passage it, clean it, passage it in the lab, it loses its virulence. It cannot cause the disease anymore. But it can go into the human cells, and the human cells can recognize it, and the human immune response can trigger an immune response. Like immune system can trigger an immune response against this inactivated vaccine virus. That's the whole premise of this. That's the whole technology of this vaccine. And as you can see, the negative controls before dose, there are no antibodies in the human subjects. But once the immunization was given, the second dose or at the third dose, the antibodies titers started increasing. So this is a pretty decent vaccine. It's an inactivated virus. So, but the thing is, how much inactivated it is, how much can it regenerate like an active virus once we take it is up in the air. So we have to go with an element of luck here and a lot of uncertainty. But there are a lot of vaccines that are actually given like that. The BCG vaccine for tuberculosis we get is an inactivated strain of BCG. So it's not that inactivated viruses or inactivated bacteria have not been used as vaccines before. They have, but it's always a chance. This is being done very fast. So if they isolated a strain from a patient, that must have been sometime in October, November of last year, and we are in September of this year. So within 11 months, they passaged the vaccine, they found the right formulation, they immunized the people. So it's really kind of a very strong race. We really are not certain how well this can be actually administered, how well it will work. But as far the clinical trials are concerned, it shows a very strong antibody titer, and the antibody titers are neutralizing antibodies as well. So that's a good sign. So as I said, that is the status of all the COVID vaccines that we have. I gave you the Serum Institute of India altered BCG vaccine, the Moderna vaccine. I gave you an example of how the inactive virus vaccine works. Then AstraZeneca, Zydus Cadila, and the Novavax, the matrix protein. So as I like this quote a lot, the more desires you have, the more of them will come true. The most important thing is you wish. So let's wish, like out of 190 vaccines, at least 50 or 60 vaccines are positive and safe, so that at least we can pick and choose what is best for a country like the US, what is best for a country like India, or what is best for an economy like other poorer economies or other places where cold storage is not possible. So I really wish that at least one vaccine from each flavor is successful so that various economies can benefit from it. So, Degnaya, it's whether it's going to be Zydus or the Covaxin from Bharat Biotech. So I end my talk here. Thank you very much for the opportunity.
Thank you very much, sir.
Thank you.
I request
any questions I can ask?
Yes, sir.
Sorry?
Hello.
Yes.
Hello, sir. Good morning, sir.
Hello. Hi.
Yes, sir. Sir, good morning. I'm Dr. Rajan speaking from Chennai.
Hi, sir.
So you said that, yes, sir. Thank you, sir. Sir, you said about the liposome-based delivery of vaccine. Am I correct?
Yeah. Yes, yes.
That's modern. But I have also heard that there is also a problem of phagocytosis of this phagocytosis of these particles. Is it so?
So yes, so this is the thing. Liposomes can actually create some sort of an inflammatory response in the human body. So that is always because the lipid, along with the soap structure and the cholesterol structure, can be very inflammatory in the human body. But the thing is, once in the when the phagocytosis occurs, the phagosome actually reduces the pH. It becomes very acidic. So it will actually break down the lipids. When it breaks down the lipids, there is a chance that there is only a certain amount of the RNA or the protein will come out of the phagosome and actually create, then the machinery kicks off. You are right. It can be phagocytosed, but there is always a way for the lipid nanoparticle to escape the phagosome because of the pH changes and how the RNA can escape outside. As you can see in the clinical trial, actually there was a decent, robust immune response there. So the dosages are important. Like that's probably the reason the dosages are important, because that's one of the reasons why they use 5 micrograms, 25 micrograms, and 100 micrograms. That's a lot. 100 micrograms is a lot. So they are running with a probability that maybe 50% of the liposomes may not be released out of the phagosome.
Okay. And I think the another reason for using this technology is for the prolonged or sustained release only, na, sir? Sustained vaccine delivery.
Yes, because it's a very sturdy molecule. I mean, it looks like an air balloon, but liposome is a pretty sturdy material. So it's like a slow release is the hope, and that's one of the reasons. Whether it's a liposome by Moderna, they try to see how far into 50 or 60 days after vaccination if they still have the antibody titer.
Okay. So,
nice question,
sir. Actually, yeah, thank you, sir. Sorry if I just missed that antibody response generated during this Moderna vaccine. So, how far this immune response is sustained, sir? In case of Moderna vaccine, is it it's a prolonged immune response is there, or how is it? How it's
So they of concept preclinical phase 1 and phase 2 in 6 months. So we really don't know how far the immune response. I mean, that's exactly the point here. We really don't know how long lasting they are. Any of these vaccines, actually, any of these vaccines, because the pandemic started in January and we are in September right now. It takes usually between 8 to 12 years to produce a stable and efficacious vaccine. 8 to 12 years. Resources in hand, and we are only 8 to 10 months. So there's no way in the world we can know how efficacious this is.
Yes, sir. And
if there are many questions, participants are requested to place their questions in the chat box.
Sir, if you don't mind, I have one last question. Sorry.
Sure.
Sir, what about the mutations, sir? What is the implication of mutations in vaccine production? Because we are hearing that many mutations are happening in different strains are also there in different countries. So how they are choosing these? How they are choosing the vaccine candidate? On what basis? Because whether they take into consideration all these mutation aspects.
So the latest report from WHO, I mean, there has always been research speculation that there are the various strains of virus and all. But the latest report from WHO, if you go by that, what it says is there are 4 amino acids in the spike protein. So the whole world is focused on the spike protein because that is your entry key into the epithelial cell, right? So there are 3 or 4 amino acids which if there is a change in those amino acids, the virus replication cycle either is slower or faster. But it is not going to prevent the immune response be any different. In other words, it doesn't matter whether the virus is whichever strain you're using, as long as the spike, that's why they're using the whole spike protein, that is 120 kilodaltons. So simple mutations is not going to harm it. That's what I mean to say.
Yes, sir. Thank you, sir.
Thank you very much. There's a question as to what factors affect the efficiency of the vaccine. Efficiency of the vaccine, for example, I can give you with an example. The recombinant adenovirus vaccine. Let me, my screen is still shared, right? So let me give you an example of how here. So if you look at my screen, if you take the recombinant adenovirus vaccine, there is a 50% probability that this vaccine is going to be fantastic, but there is also 50% probability that this vaccine can be a miserable failure. I'll tell you why. We actually are infected with adenovirus all through our lives. We get simple cold, cough, sneezes, cough, diarrhea here and there all the time. And adenovirus causes us close to 40 to 50% of those simple infections, which we don't even go to the doctor. We take a Tylenol and sleep at home. So the virus is already known to the human body. So the human body is not going to actively, aggressively pursue damaging this virus. So if we are using this virus as an adjuvant and as a carrier, the chances of the immune system to have a robust response to the spike protein is kind of 30 to 40%. It's very less. That is one of the reasons why they cleverly use the viruses from chimpanzees so that this kind of a clouding doesn't occur in our human body. It's like, very vague example. If it's a Shah Rukh Khan's movie, everyone is going to look at Shah Rukh Khan. They're not going to look at other actors in the movie. Similar way, the whole body is going to look at only adenovirus. The body is not going to look at the small spike protein that the adenovirus is carrying. That clouding, that masking is important here. And we'll have to see how well the masking, how well this vaccine works. So that is one part of the efficacy. Then adjuvants are important. And the third is how immunogenic the candidate is. Here, spike protein is very immunogenic, so that should not be a problem. Except spike protein, can we use other antigen for vaccine production? People are very much trying to see if envelope protein or hemagglutinin esterase, if those two proteins can be used. Let me go back to the first slide. To that question, right now they are using this red color protein like a petal. This is the spike protein. They are speculating if the purple one envelope or the blue one membrane can also be used, because those are also important for the virus to enter the cell. And those are also pretty predominant proteins on the virus. So those are also being considered, but no one has come to that. The spike protein is the huge protein that is all over the virus. So the probability of success is more in that as compared to the smaller proteins. Why plasma therapy fails in immunizing the patients? It's because the antibody titers are not long lasting in infected patient. The antibodies are very soluble, and most likely after 3 or 4 months, the antibody titers are not strong enough. That's one of the concerns. And also, we don't really know how well the patient is cured of the virus. If every particle of the virus has left the human body, is very difficult to know. So no matter how clean you make the plasma, there is always some doubt in your mind that you are also injecting a little bit of virus into the new person along with the plasma. I hope that answers the question. Sorry if I missed anything. With how many months we'll be able to achieve a herd immunity? I don't know. Maybe 3 years or 2 years.
Thank you.
May I?
Yes, sir, please.
How we are only talking about hydrolyzing this nucleoprotein, or what you call it as, or is it a different from the spike protein is different from the nucleoprotein?
No, sir. We are only targeting it's like neutralizing antibodies against spike protein.
Okay.
That's what I mean.
Achha. Now I was just thinking that whether these hydrolyzing enzymes will be effective.
They could be, because hemagglutinin esterase on my screen, if you notice, HE hemagglutinin esterase is very similar to hemagglutinin in influenza virus. These two proteins are close to 60% identical. So it can be used, but it's always the case, right? Like if you in the virus, the most prominent protein here is the spike protein. So the chances that your immune system will see the spike protein and neutralize it is higher than any other protein there. That's the whole point.
Okay, right.
And also that is important for the initial contact with the human cell. Without the spike protein, the cell doesn't touch the human cell. The virus doesn't touch the human cell to enter the human cell. That's the whole point.
Thank you. What should be the degree of panic? Because it is almost around what degree of panic this virus is causing.
I know, but I personally feel that initially there was a lot of panic. Initially there was unfortunate death rate, but I think now scientists and physicians overall have found a way to treat patients better, like giving steroids. For example, if I go to the slide, so here the corticosteroids or dexamethasone steroids, these steroids are helping a lot in reducing that breathlessness that people are like encountering. And also these steroids are also reducing the inflammation in the stomach also. Many people are having diarrhea problems, constipation problems, nausea problems with COVID-19 infection. So the steroids are in the heart or in the liver or the lung or intestine. They are reducing the inflammation. So some of these treatments are working very well. So I am hoping that's the reason why the mortality rate is not that high. At least in India, it's not that high. For the population we have, thankfully, the mortality is still low.
Can we relate these steroids, corticosteroids, which are synthesized in our body, as adrenaline or such?
I don't know, sir. Definitely not my domain. These are definitely externally administered steroids.
Achha, fine. Thank you very much. It was your topic was with very rich content and
thank you.
Very useful. Thank you.
Sir, there is one last question from the participant. Then we have to stop over here. The question is that for how long the antibody it is going to give protection after vaccination?
We don't know that yet. So as I said, as Dr. Rajan or someone asked in the beginning, we are having phase 1, and literally on the end of phase 1 we are having phase 2, and literally at the end of phase 2 we are having phase 3. So we are playing a test match, like a one-day match. So we really don't know how long it's going to last, how long are the antibody titers. So
okay.
Dr. Pathak, I have one last question, if you permit.
Okay, please. Sir.
Yes, sir. Please.
Yeah. Hey, Praveen, this is Kamlesh. How are you?
Hi, Kamlesh. How are you?
Pretty good. So there was a news article some time ago about taking painkillers, and that is going to aggravate the COVID situation. I was never able to get the context of it. Can you shed some light on that?
I mean, I can, not to the point, but I can give you a fair guess. So, for example, if you take ibuprofen while you were here in the US, when you take ibuprofen, you get stomach upset, right? Because It's an anti-analgesic, but it's also a pro-inflammatory. So your
Right.
stomach inflammation gets aggravated. So the whole problem with COVID-19 vaccine, coronavirus infection is actually not the vaccine as such. It is the overreaction of our body's immune system. It's the inflammation. So when you are taking a pain med to that, you are adding a painkiller that also increases the inflammation. So you're adding oil to the fire.
So
That's the point.
it's like the storm that kicks in. That is a problem.
Yes, you are adding more fuel to the fire, basically.
All right. Okay. Thanks a lot, Praveen, for an excellent talk.
Hey, thanks, Kamlesh. Take care.
It was a wonderful talk. I'm very thankful to Dr. Praveen for such an
Thank you very
informative,
much.
impressive talk, sir. Now I
Thank you
request
very much.
Mr. Anandesh Pandey, sir, to express vote of thanks.
So on behalf of Department of Microbiology, Swatantraveer Savarkar College, Bid, I express sincere thanks and deep gratitude towards the dignitaries and all the participants present over here from all the corners of India, from Jammu Kashmir to Tamil Nadu and from Gujarat to Assam. There were some from Nepal and Nigeria too. I express deep gratitude towards the President of this webinar, Dr. Hemantji Vaidya sir, for sparing his time for us. I'm very much thankful to Dr. A.M. Deshmukh sir for his presence as a keynote speaker in spite of his busy schedule. I express my sincere thanks to Dr. Praveen Almuri for his excellent reach and informative speech. I express my gratitude towards all our patrons, Honorable President Dr. Surendraji Alurkar, Honorable Vice President Shri Radheshamji Lohiya, Honorable Secretary Shri Nitinji Shete, Honorable Joint Secretary Professor Chandrakantji Mule, and Dr. Kalpanatai Chaushagar, Honorable President Local Committee Bid Shri Govindraoji Varshikar, Secretary Local Management Committee Shri Mirinji Shinsburkar. I also express my thanks towards the convener of this webinar, Principal Professor Dr. S.G. Shirodkar sir, for his support and encouragement. I'm thankful to Vice Principal Dr. Lakshmikant Bayagaonkar and Dr. Rajesh Dere, who are the co-convener of this webinar. I'm very much thankful to Dr. Krishna Bhartakke, Organizing Secretary of this webinar. And lastly, I'm thankful to all my colleagues and the members of Organizing Committee and all the staff of my college. Thank you very much. And here I declare that the webinar is over. All the participants will get the link for feedback form in the Telegram group. Thank you very much.
Thank you, participants, for their active response as well as
Thank you.
participation. Thank you very much.
Thank you for the wonderful
Thanks, all
seminar,
also.
sir. Thank you.
Thank you so much.
Thank you.
Thank you, sir. Thank you.
Okay.
Thank you.
Can we get the PPT of today's webinar?
Hello.
Hello, sir. Can we get the PPT of today's webinar?
We'll have interaction with the speaker and then we communicate to you.